In this video, we're going to talk about the synapse. So the nervous system works because information flows from neuron to neuron. And the way that information flows between cells are these functional units called synapses. Now, synapses are the junctions that mediate the transfer of information from either neuron to neuron or from neuron to an effector cell like a muscle cell or a glandular cell of the body. Now, whenever we have a neuron that releases chemicals or sends information, we call this the presynaptic neuron. So it's the neuron that conducts impulses towards the synapse. And the one that receives the information is called the postsynaptic neuron. This is the neuron that transmits electrical signals away from the synapse and receives that information. So in the peripheral nervous system, this could be like another neuron or a muscle cell, even a glandular cell, but we'll still call this the postsynaptic cell. It doesn't have to be a neuron. Like the postsynaptic cell could be muscle or glandular cells as well. And a lot of these neurons function as both, because if they're going to send information along, the postsynaptic neuron of one synapse could be the presynaptic neuron of another, because it's all linked up in a long chain. So if you look at this under an electron microscope, we see this is the axon of a neuron, and you can see that the axon collaterals can branch out and form these expanded tips, which are the axon terminals, full of synapses, synaptic vesicles full of neurotransmitter that can be released directly onto the effector cell. Now, in terms of where synapses can occur, they can either be axodendritic or axosomatic. Now, axo refers to the axon, dendritic refers to the dendrite. So axodendritic synapses are between axon terminals of one neuron and the dendrites of another. However, a lot of other axon connections can also be somatic, where the axon terminals of one neuron actually synapse on the soma or cell body of another. Now, there are other types of connections as well, like axo-axonic, dendrodendritic, and somatodendritic, but these are more rare. Now, there's two major classes of synapses, which are chemical and electrical. Now, this is showing examples of chemical synapses, where we have axons of three separate neurons that are actually branching out and forming connections with this postsynaptic neuron or effector cell. So we see here that because this axon is connecting to the cell body of this postsynaptic cell, we call this type of communication point an axosomatic synapse. And because this axon is connecting to the dendrites of our postsynaptic cell, we call this an axodendritic synapse. But they can also be axo-axonic, where one axon can directly connect to the axon of another. Now, this is important in terms of their arrangement because this helps determine how information is transferred. So the farther away these axons are, these synapses are, from that axon of the neuron, basically makes it less likely for these types of synapses to potentially cause an action potential. What we find then is that if an axon is directly on another axon, it's probably going to cause an action potential with each time it communicates. However, with these ones that are more distant from the axon of this postsynaptic cell, it's possible that the currents that are generated by these synapses may not generate an action potential in this postsynaptic cell. So what occurs at a synapse is the release of chemicals, and we call these chemicals neurotransmitters. So what happens is that the presynaptic neuron can release neurotransmitters through exocytosis, and those neurotransmitters are actually stored in synaptic vesicles. Now, the receptor region on the postsynaptic neuron's membrane has receptors, and it receives the neurotransmitters from our presynaptic neuron. And you usually find these receptors like on the dendrite or cell body, but we just saw in the last slide that they can also be on the axon. Now, the two parts of these cells, interestingly enough, are actually separated by a very small space called the synaptic cleft. And electrical impulses are changed to chemical across the synapse and then back into electrical again. So it's an action potential that causes the release of neurotransmitters or chemicals, and those neurotransmitters can generate electrical currents in the postsynaptic cell. Now, transmission across the synaptic cleft occurs in several key steps. We find that the synaptic cleft prevents nerve impulses from directly passing from one cell to the next. There's chemical events that are occurring, and this depends on release, diffusion, and receptor binding. And the reason why this is actually more regulatable is because there's lots of steps and it involves chemicals. There are things that the cells can do to either enhance or diminish their sensitivity to these chemicals. Now, this also ensures unidirectional communication between our neurons. If they're separated by space, and the only thing that can communicate between that space are chemicals, it's far less likely that one neuron can excite itself. So information transfer occurs in 6 key steps here. We learned about this to some extent going back to the muscular chapter, and so this will be a nice summary of that. Now remember, the action potential travels down your axon, eventually reaching the axon terminal of your presynaptic neuron. Now, the arrival of this action potential triggers voltage-gated calcium channels to open, and that due to its electrical chemical gradient, calcium actually flows into the axon terminal. Now, once calcium entry occurs, this actually triggers the release of synaptic vesicles, because calcium causes synaptotagmin protein to react with snare proteins that cause the fusion of your synaptic vesicles with the axon membrane, and the fusion of the vesicle actually triggers the exocytosis of this neurotransmitter into the synaptic cleft. Now, at higher impulse frequency, you release more vesicles, and this actually leads to a greater effect on the postsynaptic cell. Now, once those neurotransmitters are released into the synaptic cleft, we see that they can diffuse and bind to specific receptors on the postsynaptic membrane, and these are often chemically gated ion channels. Now, the binding of neurotransmitter opens these ion channels, which can create graded potentials, and these graded potentials can be either excitatory or inhibitory, depending on whether they depolarize or hyperpolarize the voltage. Now, binding causes receptor protein to change shape, which causes these ion channels to open, and that these ion channels can allow certain types of ions to flow. Like some ion channels might only allow sodium to flow, others might only allow chloride to flow, and this can help us determine whether it's going to be excitatory or inhibitory in terms of its effect on the voltage of that cell. Now, neurotransmitter effects are terminated by 3 key phases here. Now, our presynaptic cell can actually reuptake that neurotransmitter, and reuptake is a way to recycle those neurotransmitters. Otherwise, we can degrade them with enzymes, and this is what we saw at the neuromuscular junction with acetylcholinesterase. It actually breaks down acetylcholine by degradation. Or the neurotransmitters just diffuse away from the synaptic cleft, and this occurs within a few milliseconds. So we're saying that the effect of neurotransmitters is rapid and brief. Now, just to summarize this process, we see that the action potential can travel down the axon. Once it reaches the axon terminal, it can trigger the opening of these voltage-gated calcium channels. Calcium flows into the axon terminal through its electrochemical gradient, and then once calcium enters, it actually interacts with synaptotagmin, which then causes these synaptic vesicles to fuse with the membrane of the axon here. Now, fusion with the membrane is actually what exocytosis is, and so we see that these neurotransmitters are now in our synaptic cleft, where they can bind to receptors on the postsynaptic cell's membrane, and once these neurotransmitters bind to the receptor, this might open an ion channel. Now, if this were like the muscle cell, we learned how neurotransmitters allow for the acetylcholine receptors to open, and that allows for, the influx of sodium and a little bit of efflux of potassium, but that's what created the end plate potential, which was essentially this depolarizing current that might trigger an action potential in the muscle cell. Now, what's interesting though is for the rest of the nervous system and like with other neurons, it's not always excitatory. What can happen is that sometimes these neurotransmitters can bind to receptors that allow for ions to flow that actually make the voltage of that cell even more negative than rest, which is actually going to inhibit that cell from potentially generating action potentials. So then we call that an inhibitory type of graded potential. Now remember, the neurotransmitters are removed in 3 key ways. We can either reuptake those neurotransmitters back into the presynaptic neuron. Those neurotransmitters might just diffuse away into the extracellular fluid, or they can be broken down by enzymes. Now, there's actually a synaptic delay because it takes time for neurotransmitter to be released, and it takes time for that neurotransmitter to bind to receptors, and it takes time for those receptors to allow for ions to flow in order for the postsynaptic cell to receive a response. And this can be anywhere from 0.3 to 5 milliseconds. I mean, this is a really, really short period of delay, but there is a delay that occurs, and this can be a rate-limiting step of neural transmission. So that transmission of the action potential down the axon can be very quick, but because the synapse is more slow, this ultimately causes slowing of action potential and information spread. So we find is that with neural networks that have many synapses, they're going to be a little bit longer in terms of information spread than neural networks that have very few synapses. You know, when we talk about reflexes later, we'll learn how when it's a multisynaptic reflex, those reflexes are going to be longer because it takes longer for that information to get to, you know, either the brain or the effector organ. Otherwise, if it's like a monosynaptic reflex where there's only one synapse in the whole network, we find that it's actually the fastest type of response. So although these types of synaptic delays aren't noticeable to us, they're still very fast, and they can be experimentally determined. Now, this other type of synapse is less common, but we call this an electrical synapse. And so some neurons can be electrically coupled. And what we mean by this is that instead of having the release of neurotransmitter, the presynaptic neuron is electrically coupled with the postsynaptic cell through gap junctions. And so we're saying that the cytoplasms then of these two cells would be, you know, communicated. And so communication in electrical synapses is very rapid, and it can either be unidirectional or bidirectional because ions flow through the gap junctions based on their electrochemical gradients. And so you find these electrical synapses in some brain regions that are responsible for really rapid types of information transfer, like eye movements, or the hippocampus, which is involved with emotions and memory. And the most abundant type of synapse you find in embryonic tissue is the electrical synapse. And this is because of just rapid information transfer. However, in adults, you find that electrical synapses are more rare. However, they are faster than chemical synapses. And you might wonder, well, why do we even have chemical synapses if electrical synapses are quick? Well, the problem with electrical synapses, there's a trade-off here, is that yes, they're fast, but they're so simple because all they are is just a tube that connects one cell to the next. These electrical synapses aren't as regulatable. We'll learn about later how chemical synapses are more modifiable because you can change the number of neurotransmitters being released, you can change the number of receptors on the postsynaptic cell, you can change the amount and activity of the degradation enzymes or the reuptake of those neurotransmitters. And so those chemical synapses can be modified and molded more dynamically, whereas electrical synapses are really basic. So you might not get the same type of modifiability in the nervous system if all of our neurons and all of our synapses relied on electrical synapses.